Hello, everybody, and welcome to our presentation about the research article, Cryo-EM Structure of the Entire Mammalian F-Type ATP Synthase.
Our machine, ATP synthase, is composed of an F1 F0 membrane, which harvests energy from the proton motive force in a rotary mechanism to perform ATP synthesis. In addition, the F1 F0 membrane forms rows of dimers along the highly curved cristae ridges and plays a role in the formation of the permeability transition pore, or PTP, which triggers cell death. Prior to the article, the structures of some ATP synthases have been determined using crystallography. However, in a previous porcine or pig model, the resolution was very limited, so the atomic model for most of the membrane domain remained unknown. Detailed knowledge of the F0 domain is important because this is where proton translocation occurs and where monomers interact to form physiological dimers. In this article, the atomic models of the membrane F0 domain in the entire mammalian or ovine F1 F0 was determined using cryo-electron microscopy. This provided a detailed proton translocation pathway in mammalian F0 and key intermonomer contacts in F1 F0 multimers.
To generate a cryo-EM profile of ATP synthase, the researchers had to first purify the enzyme. They did so in 4 steps. First, they extracted bovine cells containing ATP synthase. Next, they used an anion exchange column to bind and isolate the F1 F0 complex. Third, they used an SDS page gel to identify whether ATP synthase was in the monomeric or tetrameric form. Finally, they used a coupling reaction, NADH oxidation, with ATP synthase to verify the purity of their sample.
The first step in cryogenic electron microscopy is purification and negative staining of the sample. Next, the sample is frozen and 2D images of it are captured using an electron microscope. Multiple images are taken at different tilts or angles and then compiled to create a near atomic resolution 3D rendering of the biomolecule. In general, better or higher resolution structures are rendered at lower angstroms. The highest resolution achieved by a cryo-EM device is about 1.2 angstroms. The benefit of freezing samples is that it helps mitigate the effects of electron radiation damage and allows biomolecules to maintain their native activity and functional state, including post-translational modifications. Furthermore, this allows for multiple conformational states of the molecule to be captured in one experiment. Samples are frozen by plunging them in liquid ethane and cooling them with either liquid nitrogen or liquid helium.
As was mentioned previously, ATP synthase was collected in 2 datasets, the monomer and the multimer fractions. In this experiment, the F1 F0 maps were improved and refined to an overall resolution of 3.8 angstroms compared to the previous 7 to 8 angstrom resolutions of the bovine enzyme. Focusing on F0 classification of particles in all rotational states revealed that the majority of particles classified into one consensus class, producing 3.8 angstrom map. This map was well resolved at the side chain level in all F0 areas, signifying that during rotation, the stator does not change conformation, while the C-ring stops at identical positions in each of the 3 main states. The most substantial improvements in the F1 F0 model were in the F0 region. The previous porcine model carried many errors, including misplacing the 6.8 pi subunit and misassigning helices containing subunits D, G, and E, which were also misbuilt as polyalanine. The new F0 model, which was used with existing F1 models, modeled the entire F1 F0 and included all 28 subunits. This is the structure of the F1 with all the subunits.
First, researchers analyzed the F1 subunit. This is a well-characterized structure and is known to hold a catalytic domain to perform ATP synthesis and ATP hydrolysis activity. The F1 domain resides in the matrix side of the inner mitochondrial membrane, with the rest of the structure sitting within the membrane itself. Cryo-EM data in previous studies yield very good resolution of the structure as seen by this resolution heat map. Viewed from the top, we can easily see each alpha or beta subunit with their bound ligand. Cryo-EM density studies reveal that the binding site for ADP, ATP, and magnesium ions sits in the beta catalytic subunit, right at the interface of the non-catalytic alpha subunit. Each 360-degree rotation of the gamma subunit takes each beta subunit through 3 conformational states, beta TP, beta DP, and beta E. This consequently hydrolyzes 3 ATP during counterclockwise rotation and synthesizes 3 ATP during clockwise rotation. No magnesium ions were originally present in solution, yet ADP and magnesium ion densities were found in each alpha subunit. ADP density without inorganic phosphate is found in beta TP and beta DP subunits, but not in beta E. The beta E subunit was found to be dramatically opened by the rotation of the gamma subunit and is effectively empty. Due to the beta TP and beta DP being bound by ADP without inorganic phosphate, the enzyme composite was most likely in a state of magnesium ADP inhibition. The OSCP subunit at the F1 crown forms tight interactions with the alpha and beta subunits of F1. This interaction strongly but flexibly attaches the F1 subunit to the peripheral stalk made up by subunit B here in blue. The peripheral stalk is anchored to the inner mitochondrial membrane via interactions with the transmembrane helices of subunit B and ATP8. The type of flexible binding of the peripheral stalks allows for a slight wobble of the F1 subunit during rotation. Rigid body fitting of the subunits into maps of other rotational states allowed researchers to model the movement of the enzyme during its catalytic cycle. Here we can see how the entire F1 subunit wobbles slightly with each catalytic rotation. Additionally, we get a good visualization of the two main rotational subunits of the enzyme, the C-ring alpha helix cluster and the gamma subunit, along with the visualization of how the peripheral stalk stretches and accommodates for the wobbling of the F1 domain. Next, the researchers analyzed the F0 domain of ATP synthase. This subunit sits in the inner mitochondrial membrane itself and can be divided into three main regions. First, we can look at the rotating C subunit. Next, we can look at the proton translocation cluster consisting of subunit A, DAPIT, 6.8pi, and ATP8. And then finally, we can look at the hook apparatus, which consists of subunit F, G, and E.
Resolving the F0 structure enabled the authors to discover the connection of subunit E to the lipid plug in the C ring. Specifically, subunit E, also a part of the hook apparatus, is connected to the intermembrane space lipid, ISL. The lipid plug consists of two lipid chambers, the ISL and the matrix lipid MSL. The two chambers are separated by a valine on subunit C. Unlike the MSL, which is a rotating double acyl chain, data suggests that the ISL is a single chain lipid that does not rotate within the C ring. Although the lipid type is unknown, the cryo-EM averages suggest that rotation is lipid structure dependent. Finally, the data shows that the flexible stalk is anchored by subunit E, which is attached to the C ring, and to the lubricated lipid plug, which allows for rotation at the connection points between the F1 and F0 via the gamma subunit.
Researchers used cryo-EM data to propose a chemical pathway that explains proton movement across the membrane via F1, F0 ATP synthase. Subunit A has a unique belt-like horizontal hairpin that matches shape closely with the rotating C-ring and helps ensure smooth rotation and subsequent protonation and deprotonation steps. The sequence is proposed as the following. First, a glutamate on subunit C is protonated from the inner mitochondrial space. Then subunit C rotates and moves the protonated glutamate, and in order to prevent enzyme slipping, it is checked by arginine 159. This residue only allows the glutamate to pass if it is protonated and stops unprotonated glutamates from completing the rotation. A different glutamate on subunit A then deprotonates the subunit C glutamate, allowing for the proton to be passed through the exit half channel by way of conserved amino acid residues.
From the discussion of the PTP, the researchers moved on to discuss the dynamics of F0-F0 interactions. They created two hypotheses to model interactions between F0 complexes of ATP synthase. They're called the on-the-cristae ridge hypothesis and the neighbors in a row model. In the neighbors in a row model, subunit A and subunit C are interlinked by protein factor IF1 and interact with each other in the presence of DAPIT, a linkage factor, as shown in figure E. Together, two DAPIT, two G and E units from the F0 structure form the core of the tetramer architecture and allow the proteins to sit at the edge of the mitochondrial cristae, as shown in figure F. In the on-the-cristae model, opposing F0 monomers attach to each other to form a covalent interaction, as shown in figure C. This experiment proves that the on-the-cristae model is correct, because the removal of any one of the following components, DAPIT, the G, or the E subunit, kills the dimerization/tetramerization process, but all of these components are necessary for the ATPase to sit on the edge of the cristae, and all of them happen to be present in the in vivo sample.
Lastly, the authors proposed a permeability transition 4 or PTP opening model. The authors first observed that the binding of calcium to the beta subunit of F1 elicits changes in the F1-F0 structure. Steps 0 to 3 possibly represent the stages of PTP opening as the synthase gradually disassembles. In step 2 and 3, the C ring is unobservable and thus omitted. They were then able to propose that the PTP opening is facilitated by the binding of two proteins to the OSCP and the F1 complex. Cyclophilin D, CYPD, increases the F1-F0 sensitivity to CA facilitating pore opening. And cyclosporin A, CSA, not pictured, binds similarly but inhibits the PTP. In their model, CYPD binds to the crown and stiffens the OSCP-PS assembly. almost like a molecular grip. This grip, coupled with the large calcium-induced F1 conformational changes, would result in pulling or pushing forces that will lead to subunit E pulling the lipid plug out of the C ring. Water would then rush out of the matrix and further disrupt the buried hydrophobic core of the C ring.
Resolving this structure allows for ATP synthase data interpretation, as the researchers were able to propose a PTP model for apoptosis and show that subunit E's double role is the second anchor point for this data, along with the OSCP's double role as a hold for the lipid plug in the C ring.
Lastly, these were some of our outstanding questions. The experiment didn't elaborate on the translocation mechanism, the F0 dimerization / crystate formation, and PTP opening / regulation. So further research on these topics would help our understanding. Our second question would be, how specifically does the F1 F0 model apply to the development of treatments for various neurodegenerative disorders? And lastly, what are the identities of the lipids in the C ring? Thanks so much for watching. Please direct your questions to Q&A.